What's good, y'all, and welcome back to another edition of Mr. Fitzgerald's biology class. Today we're going to dive into mutations, which a lot of students think is like, oh cool, we're going to talk about things with extra arms or extra eyeballs or something crazy like that. No, it's not that exciting overall. There's no going to be graphic pictures or anything like that. What we are going to talk about is the relationship between protein synthesis, which we've been working on through that central dogma, and changes in the DNA, which are mutations. So before we get into those mutations and the different changes that can happen to the DNA, let's start talking about a little review of our protein synthesis. So overall, we did go over the different codon charts. So I gave you the circle codon today for this little warm up instead of the square one, but you can use either one that you feel comfortable with for this. But go ahead and pause it so that way you can try to work this out, and then I'll work it out with you right now. So what amino acid is coded for from the DNA sequence of CAT? A lot of students will make the mistake and try to put CAT in this chart. That doesn't work because that's the DNA sequence. And remember, these are called codon charts, which means that I need a codon or an mRNA strand. So in this one, if the DNA is CAT, I need to know the corresponding mRNA here. Okay, so the mRNA that would match up with this, well, I have a C, so that's going to give me a G, an A gives me a U, and a T gives me an A. So I'm going to use this GUA when I use the chart to figure out what amino acid it is. So looking at the chart, I remember that I use the inside and I work my way out. So I have a G here, I have a U here, and I have an A, and that's going to give me the amino acid valine. Okay, so the CAT on the DNA sequence is going to code for the, or be transcribed as the as the codon, transcribed as the codon GUA, and that's going to be translated in the codon chart to the amino acid valine. All right, so let's start talking about mutations overall. This is a little bit of an old PowerPoint, so I apologize for the page 88. We don't really have page numbers this year, but think about what would happen if we had a change in our DNA sequence. All right, so if we have our DNA code that's supposed to make specific proteins, we had some change in that code. More than likely, we're going to change the protein. So let's kind of see what would happen with that. So there are different types of changes. There's 4 overall that you do have to know. The first one is the most obvious one, the substitution, right? If I'm substituting something, that means I'm flip-flopping something. So instead of having maybe an A where it's supposed to be, in this case, it's been replaced with a G. So my codons read AAT, GTG, CCG, and instead of reading that like it's supposed to, this DNA strand now reads AGT, GTG, CCG. So the last 2 are the same, but I've swapped an A and a G. Now you might think, okay, well that's just a minor thing. It shouldn't be that big of a deal, but it actually can end up with basically giving us a new amino acid. So when you put this into your codon chart and you get the corresponding mRNA that goes with it, you'd find that it gets leucine. Where down here, if you just swap that one letter, now you end up with a serine. And most of you are like, I don't really know the difference between leucine and serine. That's true, you don't. And in this class, you won't ever get into the chemical structures, but just having a change like this could actually change the shape of the protein. Remember, proteins are supposed to fold in a specific way to give it its shape to allow it to do its job. So maybe that leucine allows it to fold at a specific point, but a serine would keep it open. So now I don't have the same structure, and maybe the protein doesn't function the same way. Or honestly, sometimes these swaps out really didn't do anything, right? If it's supposed to bend, it still bends, either if it's leucine or serine. So I don't know what's happening here because I don't have enough information to go off of to see what the rest of the protein would be, but we do know that the amino acid changes at those points based on the substitution of an A and a G. All right, now there's another easy one, insertion, where you stick something extra in. So maybe DNA polymerase stuck an extra DNA molecule, or sorry, nucleotide in there, and ligase never picked it up as the editor. So the normal strand would read as such, right? So you got all these codons here, and if I insert maybe a single letter, now basically what that does is it has a domino effect. If I put one in, because it reads in groups of 3s, everything got shifted. So this is called a frame shift because it's reading differently now. So normally it's supposed to read these 3, then these 3, then these 3. But now I've inserted one extra nucleotide there. So these 3 still read the same, but everything else shifted. Instead of being CCC, now it's TCC. And that third C gets bumped to the next one, CAC. And that T gets bumped to the next one. So everybody else gets bumped as an impact. So it's supposed to be glycine, proline, purine, but instead now it's glycine, serine, histidine. Okay, so again, you probably don't really know what cysteine, histidine, thurine, and proline are. That's okay, but you can tell they're different, right? It shows up as red here, showing that it's different. So I get every amino acid after this point, most likely is going to be affected. Just because I put that one extra nucleotide in there, now everything else gets bumped to the side. If I insert maybe a set of 3, actually that's not as bad overall. So this is not a frame shift. It still reads in those groups of 3s. Nothing got bumped to the side. It's just going to have an extra amino acid put in between these two than it normally would. So there's frame shifts where basically everything gets screwed up, and non-frame shift mutations where just maybe a small piece gets added in, a little amino acid gets added in, but the whole sequence is more or less still the same, right? So the next one is a deletion. Okay, a deletion is when you remove some segment. So this could be a large piece, it could be a small piece. So instead of having all these A's, T's, G's, and C's, we have a chromosome here, right? When these might represent different genes, gene A, gene B, C, D, etc. A deletion event, C got deleted for some reason, it just never got copied. And that can happen, maybe some environmental toxin got involved or something else, or something interrupted the replication process, or whatever it may be, where DNA got messed up, okay? So if I remove that, clearly, if this gene is supposed to make a specific protein, then I've lost that protein. My cell can no longer produce it anymore, and that actually might have some impacts on the cell as a whole. It might not be able to survive, it might survive differently as a result, or it just does nothing at all because that protein was kind of pointless anyway. So there are some of those events where nothing really happens. It's just a change, but not a big deal. You can get frame shifts when you have deletions, so instead of inserting an extra letter, if I take one letter away, I've basically shifted everything in instead of out, like I did on the last slide. So instead of putting that extra T in like on the last slide, we removed 2 of the letters there. We're shifting everything back in, so then we're going to get different amino acids as a result as well. All right, and the last one is called translocation, okay? Trans means across, and then location, it means location. So I'm flip-flopping things. So this is where you have some sort of DNA literally just kind of flips between the two, but they're not supposed to. So this isn't like crossing over. Crossing over would be like an equal exchange of genes. You have these genes, I have these genes, and we flip-flop them. As the same. These would be 2 non-homologous chromosomes, like let's say chromosome 1 and chromosome 7 or something like that, just to give some different numbers here. So chromosome 1 is supposed to have these sections, these genes on it. Chromosome 7 is supposed to have these genes on it, but for some reason something collides or they get kind of mixed up inside the nucleus, and now whole chunks and sections got flip-flopped. So they're showing this end piece here gets moved down to here. Now maybe my DNA is not able to be read as efficiently. Like if there was proteins that needed to be read on chromosome 1, the cell has a hard time finding it, or on chromosome 7 or vice versa, or it might actually make it better. A lot of these mutations aren't always bad. Sometimes it actually helps the cell. Sometimes, again, it does nothing at all. So the real big idea here is translocation is just big sections get exchanged between different chromosomes. Deletion, I'm removing a section from a single chromosome. Insertion, I'm putting in some nucleotides, and then substitution, something got flip-flopped within the same sequence. Okay, so what could a small change in DNA lead to? Well, it might only change 1 amino acid. All right, so if I have a very small change, maybe I get 1 amino acid different. It might change a lot of amino acids, especially if I have a frame shift. If I inserted one piece, or if I deleted one piece, that's going to shift everything else after that point. So when the proteins or the ribosome is reading that mRNA sequence, it's all screwed up now. All right, or many do absolutely nothing at all. There's actually a lot of mutations in all of our DNA, and they don't do anything, so you wouldn't notice if they're there or not. Our DNA is just a compilation of mutations anyway, because you and I look different. Therefore, I'm making different proteins than you to give me my different physical appearances and traits. So we all have different mutations within us. No one is a, quote, perfect person. Our DNA is all kinds of messed up, and that's a good thing because it gives our species a lot of survivability. But a lot of cases, we have sections of DNA that are all messed up, but they don't really make a protein, so it doesn't really matter. All right, so let's talk about that one. So how many nucleotides are in human DNA? We got about 3.2 billion nucleotides in our DNA. That's a lot of nucleotides, so there's a lot of room for error. We also have somewhere between 30 and 40,000 genes. We're still kind of working on that number a little bit. So basically, more or less, we have the ability to make 30 to 40,000 different proteins. within our DNA sequence here. Now, how much of your total DNA actually makes a protein, how much of them are genes that actually make proteins? Actually, it's only about 1%. 99% of your DNA actually does not code for a protein, it is just there. For the purposes of this course, we can consider that filler. If you take AP Bio or something else, then you'll realize that it's a little bit more intense than that. There's things that turn on certain genes and turn off certain genes. We don't care in this class. In this class, 1% of your DNA actually codes for a protein, the other 99% is just there. So if a mutation happens and it's in that 99%, you wouldn't really notice the difference. Only if it's in that 1% of DNA. So there's a good chance that you have all kinds of mutations happening inside your DNA every time it's being copied, but you really never see the effects of it. All right. So will a mutation impact a protein or an organism? That really depends. Most likely not, because again, 1% of the DNA actually codes for a protein, so those would have to be impacted first. However, some of those mutations can actually, make some changes, but it all really depends on what amino acid got inputted, what protein was affected, how many amino acids were affected. Sometimes it make the protein good, so some mutations actually make the protein better at what it was doing than before. Some make it worse at what it was doing than before. So not all mutations are innately a bad thing. They're just differences in the DNA, all right? So that's all I have for today. Make sure you work on your exit slip and message me if we have any questions, and go back and review some of the videos because we do have a test coming up and you want to be prepared for that. But that's all I have for today, and have a great day.